As Liam said, my name is Mike Robson. I'm chair of GCSE Biology here in the UK. So I'm responsible for a lot of our own domestic exams, and I do some of the training for the International GCSE Biology as well. Here in the UK, it's a very wet and grey morning, about 9 o'clock in the morning. I know you are from various parts of the world and no doubt at various times of the day. So our plans for today are to basically go through the specification, and if you're familiar with the old linear exam, then you realize there's now some modular options as well. So we'll spend a little bit of time on that. We'll look at the different assessment models. We'll then go through the specification and the content and discuss some of the points of that. Look at the assessment objectives. Look at the taxonomy and command words. Look at the core practicals. Look at the math skills. And then we'll finish off in 2 hours' time or so, looking at the support that is available from Pearson's. As Ian said, please use the chat room. I'll go through a little exercise next just to ensure that you get a chance to do that. And then we'll talk about the various issues. I'll try and keep my eye on the chat and pick things up as we go through. So today we'll identify how the qualification is devised and some of the fundamental sort of documents that are involved with it. We'll review the content of the qualification, explore how to plan the course and the lessons, understand the assessment of the qualifications and how to prepare your students for it, and to identify support from Pearson's. So just as a little sort of exercise to make you familiar with the chat room, I'd be interested to know how many of you are actually brand new to this course and are teaching it, as opposed to some of you that have perhaps had students that were entered last year. So perhaps the easiest way in chat is if you could simply respond by saying, I'm new, or I had some students last year. And then if there's anything special you're hoping to get from today, then please put that in the chat as well. I've outlined what we're going to do, so don't bother repeating that. But if there's something else, then please put that in the chat room now or as we progress through the course. I can see we've got quite a lot of people in the room, and almost everybody's coming in. I think you can all see the same chat that I can. So you'll see that the vast majority are coming in to say they're new to the course. Having said that, I've got Amand that's just come in to say they're not new. But the majority are definitely new to the course. So thank you very much for that. And you're now familiar where to put things in chat. I'll be asking you for your opinions on some things as we go through the course. So just a little bit of information about Pearson's. They're obviously a huge organisation. In fact, the UK's largest awarding organisation, and they go back some 150 years. And actually, there are over 150 million, 3.5 million students that are actually working on Pearson qualifications around the world. So a very big organisation, with a lot of history behind it. About 6,000 or so schools and colleges are directly involved with us in our courses. So it's a great organisation that can support you in what you are doing. So let's look at the actual specification itself. In the resources is a copy of the spec, which you are able to download, but equally you can get the specification from the Pearson website, which I'll show you in just a minute. You might or might not notice that the specification has on the front issue 2. The issue 1 is the one that came out originally. If you're working from issue 1, don't panic. Issue 2 was just really correcting a few little bits and pieces, changing the wording about the assessment objectives and things like that in a couple of places. The actual content that you have to teach and all the key information hasn't changed. So don't panic if it says issue 1 on the copy of the specification that you've got. There is also now the modular specification, which gives details of the nature of how the modular assessments work. But the actual specification bit, the bit that you teach, is identical to both. So as long as you have one copy, I personally prefer a printed copy to sort of look things up, but of course it can be available online as well. And that's your sort of Bible to what to teach the children as you go through. So you can find that specification here on the Pearson qualification website. If you just type in, Pearson qualifications, you'll see everything comes up. Then you go to the International GCSEs, and then you obviously, for today, you would select biology or double science, depending on what it is that you're doing. And you can see you can download the specification from there. This is an original slide. It says, specification and sample assessment, there's 3 available. The exam materials, it says 13 available. I think it's something like 180 something available now. And I'll show you the slide again later on when you see those numbers are being updated. But that's the generic place where you go to find support material for your teaching. So what does the specification contain? Well, it tells you, first of all, the assessment model, exactly how the marks are allocated, what the topics are that are tested in the different styles of different papers, the styles of the question paper, how paper 1 may differ from paper 2, and it also sort of gives you the breakdown of how the assessment works on each of the different exam papers. The specification will also obviously give you the content. So blow by blow, when we are writing the exams, we sort of have to list and say, we think that this question is assessing spec point 2.12, and we have a debate. And if anyone thinks it's sort of gone outside the boundaries of that spec, then that question can't be allowed. So the assessment is very much based on the spec. And then we have assessment objectives. These are assessment objectives 1, 2, and 3. And we'll be looking at those in a little bit more detail in a few minutes' time. The command words are the word that obviously starts a question. So it may be describe, state, explain, discuss, compare. And these command words, again, we can look at those later in this course this morning. And there's a module 2 next week where we will go into even more detail about things like that. The maths, all GCSE sciences have a certain percentage of maths marks, and so it makes it very clear what the maths skills are. And if you are teaching just biology, then it's important that your students realize that obviously these maths skills are common to chemistry and physics, and of course to maths itself. So you may want to talk to the other teachers to ensure that you're not sort of confusing the students by going with one style of maths compared to a different style of maths in terms of what they're learning. Practical skills. The practical skills again are common through chemistry and physics. Each of the subjects has a set of core practicals which are detailed in the specification, and without doubt the best way to prepare those students for the practical skills questions is to actually do the practicals hands-on, live in the classroom. That's what makes science different to history or geography or whatever, in that we can investigate, in biology, real living materials, plant materials, even animal materials, and following through. And through all of this runs the concept of transferable skills. And I think the most important thing there is certainly, changes that I've seen in my lifetime, and that when I was a student way back many, many years ago, then you would be able to sort of look things up in an encyclopedia or whatever, and that was it. But increasingly now, there's a whole host of different media, and you have to be able to sort of assess what you're reading and examine, is what I'm reading correct? Is it not correct? Are the sources reliable where this information is coming from? And those sort of skills to be able to do that are core to the sciences, and they are the transferable skills that are not just to get through a GCSE biology exam, but are useful life skills in terms of the business of sort of saying, well, does that information, is it based is that practical information based on an adequate base of knowledge? Is it based on evidence? What is that evidence? How is that evidence being interpreted? Those are the transferable skills that are increasingly important today and are throughout the questions and the syllabuses that we teach. So let's look in more detail at the actual assessment itself. So you now have two choices with this specification, one of which is the original, which has been with us since 2018, and the second is new this year in 2024. So the choice now is linear or modular examinations, and I'll explain those to you exactly as we go through. And you are free to choose whichever of those particular models you think is best for your students and your school and your teaching. There are no separate practical exams. Practical skills are assessed on both papers through questions based on the core practicals, but also wider than that, so the knowledge gained in the core practicals can actually be applied to a wider context. The papers have similar question styles, but paper 2 for linear has some additional content, and I'll explain that to you now. And there is no tiering. In other words, we don't have a foundation paper and a higher paper. Here in the UK, the foundation paper is targeted at grades 5 to 1, and the higher paper is targeted at grades 9 to 4. But here, the full range of grades are available on the papers. Okay, so what's this new modular route all about? Well, let me stress straight away, the teaching, the specification is exactly the same. It's just the way that information is carved up in order to sit the exams. So the modular route, the unit assessment can be taken over multiple exam series. Grades are calculated on raw marks, which are then converted to what's called a UMS. Students can resit individual units at any exam series. And once a student has all their unit results, they can cash them in for these results. And the modular route is new, and I think Pearson's are the only international GCSE that are offering that modular assessment. The way it's been since 2018 is the linear route. So the assessments for all units are taken together in one exam. So you will teach the course for 2 years. Some schools take it over 3 years. And at the end of that, there will be the exam, 2 papers on all the content. The grades are calculated based on the raw marks of that single exam at the end. So the students will sit paper 1, it will be marked, they will get a mark for that. They will sit paper 2, it will be marked, they'll get a mark for that. Those 2 marks will be added together, and then they will be compared to the results from previous years to see if the exam this year was perhaps a little on the harder side or a little on the easier side, or other circumstances that may have affected the results. And that mark will eventually be changed to a grade 9 to 1. Obviously, students can come back the following year and resit, or in the November or the June exams, come back and resit the whole thing again if they choose to. There's no rule that says they can't do that. But it's effectively one exam consisting of 2 papers at the end of the course. So just to stress again then, for paper 1, if you're doing paper 1 linear, then it is a 2-hour exam. It's 110 marks, and it covers about 60% of the exam. In the specification, you will see, if you haven't already, that some items in the specification are marked in bold with a letter B. Apology, it says C here. That's obviously been copied from the physics. My apologies for that. It should be the B mark that's on there. So that content is not, I stress, not examined in paper 1. The reason being that paper 1 is taken by the double science students as well as the pure biology students. And I'll show you how that adds up in a couple of slides' time. So the linear paper 1, when students are revising for that paper 1, they can ignore the bold bits, the bits with a letter B against them in the actual specification. Paper 2 we'll talk about in a minute. The modular paper 1 is a shorter exam, 1 hour, 40 minutes for 90 marks, and it covers 50% of the spec. And it will just cover topic 1 and topic 2 down to G. Topic 2 goes on a little bit further past G, so it's not the whole of topic 2. So the advantage of that is that the students are only revising half of the specification before they sit an exam on it. Once they've sat that exam, that mark is filed, and then they can go on and learn the second half of the material from then on. Okay, so that's a key difference. The paper 1, if you're doing linear, will be a 2-hour exam on all of the specification, ignoring the bold stuff. If you're doing modular, the paper 1 is on half the specification, topic 1 and A to G of topic 2. Paper 2 is a shorter paper for linear, 1 hour, 15 minutes, a total of 70 marks. And this is not sat by the double science students. And so it includes the stuff in bold, the stuff with the letter B after each of the spec points. And I say we'll look at the spec in more detail in just a minute. Okay, so the linear paper 2 is on everything, both the non-bold and the bold parts of the spec, and that is not taken by the double science students. The modular is 1 hour and 40 minutes, so compared to paper 2 linear, it's longer, but it's the same as it was for the paper 1. Again, it's covering half. And again, my apologies, there's an error here on the slide in the numbering. So that's looking at topic 2, the remaining parts. Remember, topic 2 was parts A to G. So here are H, I, and J. So that's topic 2. Then topic 3, reproduction. Topic 4, ecology. And topic 5, biological resources. Okay, so a slight error on that slide. My apologies for that. Okay, I'm aware of quite a lot of things in the chat. I think quite a few of them are technical, asking for help. So I'm hoping Leon will pick those up. My apologies if I've missed a biology one. It's difficult to sort of present and pick up what's in chat as well, but I'll do my best to keep an eye on it. Okay, so here's a copy of a page from the spec. This is, as you can see, it's actually the part H on transport, and this is a bit on humans. So you can see there, it says exactly what it is you're supposed to teach. Describe the composition of blood, red blood cells, white blood cells, platelets, plasma. So that could be examined in paper 1. It could be examined in paper 2. If it's modular, because it's H, it will be in the second unit because it's after G of module 2. So you can see there 260, 261, and so on down. When you get to 263B, that's got the letter B on it, and it is in bold. That means double science students don't have to know that. And because the double science students do paper 1, then that topic will only be examined on paper 2. So if students are studying for their exams and you are doing the linear course, then for paper 1, you can ignore revising those bold bits. And of course, proportionally, that means there will be more of the bold bits on paper 2 because the bold bits are only on paper 2. So it's a good idea to really concentrate on the bold bits when revising for paper 2. So that's how the spec is laid out. So I said with the modular way then, when you're looking at that, you may want to change the way you teach by using the GCSE modular. We'll support your planning. These have been produced now, so you'll find them on that web page I showed you earlier. There are course planners. I think the schemes of work are probably identical because it doesn't actually affect it. It's just how you break them up. And there are various guides how to do it. So the first teaching of the GCSE Biology modular is now, and the first exam, so there'll be a module 1 exam and a module 2 exam in May, June 25. After that, they'll be in November and June. And as I say, it could well be, once that's fully established, that you teach module 1 and sit it, say, in November, then you teach module 2 and sit it in the June. And if you wanted to repeat module 1 in the June to try and get a better score, that option would be available to you. So the resits with the modular, okay, so the learners can resit any unit irrespective of whether the qualification, pardon me, is to be cashed in or not. If a learner resits a unit more than once, only the better of the 2 most recent attempts at that unit will be available. So just to stress out what that means, if the student takes the exam in unit 1, fine, they get a mark. They take unit 1 again, they get a mark. The better of those 2 marks will apply. If they sit it again, it seems unlikely, but if they sit it again a 3rd time, then that first mark will be dropped and it'll be better of the remaining 2 marks that will go through. The results of the units will be held in Pearson Unit Bank for as many years as this spec exists. And then once the International GCSE Biology, sorry again for the error in the slide, biology module has been certified, then it'll count towards further award. You can't use it for further awards for the same qualification. So once you've cashed them in and said, that's it, you can't say, oh, I've changed my mind. I'll sit that unit again and see if I can get a better grade. Once you decide you've cashed them in, then you've cashed them in. So just to summarize what I was saying then, so when you take biology paper 1 and biology paper 2, you have a GCSE biology qualification. And that's the same for chemistry and it's the same for physics. However, you can enter for double science, in which case you just take the biology paper 1 and you've only learned the stuff that is not in bold in biology. Ditto for chemistry, ditto for physics. You cash in those 3 qualifications and that is your double award science worth 2 grades. Okay, so the basic structure is there for the exam, and that's why you have the bold material in the spec, because that's obviously important to whether or not you're doing double science. It makes it the standards are the same within the double science as it is within the biology or chemistry or physics. Okay, so that's the various assessment models, a bit more complicated than it used to be because of the modular options, but those are available to you. And therefore, one decision for you to make, you know, coming up in beginning of next year is, do you want to cash in using the modular approach, or do you want to stick with the linear approach? Okay, and obviously there'll be support available for you if you want to come through. There is also a single science award, i.e. a subject. I see one of the questions in chat there. There is a single science award where you obviously do a smaller, it's still the same spec points, but a smaller reduction. It's a separate exam altogether. It's not part of this structure that I've explained to you today. So students can do just a single science GCSE, which is bits of the existing biology, bits of the existing chemistry, and bits of the existing physics. Okay, so looking at the topics then that are in the thing, what you'd reasonably expect in the sort of specification. So the first topic in the specification we'll look at in a little bit more detail, but they summarize them there. So you've got the nature and variety of living organisms, the structure and function in living organisms, which is the big one, the traditional sort of, that's what a lot of, you know, all the chunks of what people regard as the core biology is in there. Reproduction and inheritance, increasingly important now as we look at the impact of genetics on our food production and understanding of disease and so on. Ecology and the environment, again, very relevant now, you know, with global warming and some of the other factors that are involved, so an understanding of that. And then using biological resources, crop plants, fish farming, genetic modification, cloning, some of the issues that go on there. So those are the charts. So let's look at them in a little bit more detail. So topic one, the first, again, sort of what might be regarded traditionally as the introduction to biology. So it's subdivided into two areas: the characteristic of living things. That's just basically one spec statement that lists through, nutrition, respiration, responding to the environment, moving, reproduction, growth, and so on, as you'd expect. Focuses on the classification of organisms. So it looks at the variety of organisms and looks at plants and animals and fungi and protists, and looks at the sort of describing the common features shown by prokaryotic organisms such as bacteria, and then understanding the term pathogens and the known pathogens, the fungi, the bacteria, viruses come into that. So a sort of chunk of introductory stuff, possibly something that you'd introduce fairly early on in the course. And it's going to constitute about 5% of the marks. But of course, it reoccurs. So understanding words like pathogens, it's understanding the basic principles of living things, the respiration, the nutrition, the growth, the reproduction, the sort of core to what biology is all about. Topic two, the big one. Okay, so lots and lots of stuff. I think it goes on about 4 or 5 pages in the specification. So this is the big sort of chunk, the structure and function of living organisms. So it's looking at what an organism is. So looking at organelles, cells, tissues, organs, systems. Looking at the structure of cells. So again, making sure the students are familiar with the terms, cytoplasm, cell membrane, cell wall, mitochondria, chloroplast, the basic sort of organelles that would need to be understand, the difference between animal and plant cells. Then the sort of biological molecules, the understanding carbohydrates, proteins, lipids, and there's a practical in there, investigating the presence of glucose and starch and protein, the standard sort of food tests, understanding about enzymes and how they're catalysts, the effect of temperature. There's another core practical in there, looking at enzyme activity. So an important topic on biological molecules with 2 core practicals in there. And in fact, a third one, which looks at pH, is in the paper too, the bold part of that spec. Movement of substances, again, core to understanding, understanding about, the area to volume ratio, distances that things can move, temperature, concentration gradients. And of course, there's a core practical in there as well on diffusion and osmosis. So an important section there. Nutrition, as you'd expect, and that's split into the flowering plants. So looking at the process of photosynthesis, the equation for photosynthesis, the structure of the leaf. And again, there's a core practical in there involving photosynthesis and the production of oxygen from the leaves. Then the human nutrition, the digestive enzymes, the role of bile in the digestive system, the use of vitamins and the use of the different vitamins. They're all sort of parts of that energy levels, general nutrition. And of course, there are around the world some sort of obesity issues in terms of ultra processed foods, which are relevant to the human nutrition. Respiration, understanding the standard respiration, understanding the word ATP and what that does, aerobic and anaerobic respiration. And there's another core practical in there investigating the evolution of carbon dioxide. Gas exchange, a lot of that is bold. Certainly all the flowering plant stuff is bold, so you can leave that for paper 2, or if you're doing double science, you don't have to do it at all. And then the human gas exchange, obviously the structure of the lungs and the thorax, the bronchi, the bronchioles, you know, the adaptations of the alveoli, blood capillaries. The diffusion stuff will come back in here again. And again, there's a core practical on breathing in humans. So that, if you're doing modular, that's where the first unit ends. So that would be a separate exam if you're doing modular. It stops at G. And then H, I, and J would be in the second module. So transport, obviously, again, that's split between flowering plants. Some of that is in bold, you know, xylem, phloem, root hairs, transpiration. The human side of it, with the blood, the red blood cells, plasma, the immune system, understanding vaccinations, platelets, the structure of the heart, increasing risk of developing coronary heart disease, arteries, veins, capillaries, fairly traditional stuff on that. Excretion, a little bit about plant excretion and carbon dioxide, but a big chunk on humans. Again, a lot of that is in bold, so it's paper 2 only, and that's the kidneys and urinary system, Bowman's capsule, you know, the sort of ADH and urine and water, urea, ions, and so on are there. And finally, for section 2, coordination and response. So again, split between flowering plants and humans. So understanding, you know, geotropism, phototropism and things in plants. And then when it comes to humans, the central nervous system, hormones, structure and functions of the reflex arc, the eye, temperature regulation, and obviously the role of hormones in the reproductive cycle come in there as well. So that's a big, big chunk of what many would regard as the sort of core biology. Okay, so a big section of specification and core principles that feed into other things are in section 2. Topic 3 is reproduction and inheritance. So that more or less speaks for itself. Obviously, reproduction both in flowering plants, you know, insect pollination, you know, seed germination as a core practical in there on seed germination, and then the human side of it as well. So the male and female reproductive organs, understanding the role of hormones, FSH, LH, the menstrual cycle, pregnancy, estrogen, testosterone, all come into that. And then the inheritance, as you'd expect, you know, structure of DNA, looking at what genes are, understanding genetic analysis using terms like dominant, recessive, homozygous, heterozygous, understanding what codominance is, that one's in bold, and then looking at mitosis, meiosis, you know, the effect of sex, offspring and fertilization, being able to trace that through chromosomes, haploid, diploid, mutations fit into what's going on there. And there's a little bit on sort of explaining Darwin's theory of evolution, and things comes in at the end there as well with natural selection. Topic 4, the ecology and the environment. Again, rather self-explanatory, I think. So organisms in the environment. There's a couple of practicals in there, looking at population size in different areas, you know, using quadrats. The topic of biodiversity, very much sort of in the press these days. Looking at feeding relationships, so, you know, food webs, food chains. Looking at cycling within the ecosystems, the carbon cycle, the nitrogen cycle. Carbon cycles for paper 1, nitrogen cycle is paper 2 only. And then, of course, the effect that humans have on our environment, understanding things like, you know, pollution, you know, increasingly now carbon dioxide in the atmosphere, how human activities contribute to that, the global warming, pollution as a result of the size of the human population, eutrophication. Well, you get some topics which are called synoptic topics, where you sort of draw together lots of things which you might want to leave towards the end of the specification. So things like eutrophication are one where you draw together lots of different bits of the syllabus to understand what eutrophication is all about. Okay, so some of that can be quite challenging, and some of it is best done towards the end of the course because of its synoptic nature. And then finally, the use of biological resources. So you're looking at food production, so obviously crop plants, but also the use of microorganisms, the use of yeast, industrial fermenters, understanding concepts like fish farming and how control of disease comes in when there's large numbers of the same species all in the same place. Selective breeding, how you sort of, end up with cows that can produce a lot of milk, sheep that may have, a different shape to them in order to develop the meat that's required in different parts of the body, all through sort of selective breeding that has come through. Genetic modification, the details of restrictive enzymes and how to cut DNA, the use of plasmids and viruses, perhaps an example of how insulin is manufactured would be part of that. And cloning, again, micropropagation, tissue culture comes into that. And how, cloning mammals, taking a diploid nucleus from a mature cell, and the ideas of Dolly the sheep and so on comes in there, transgenic animals. So look, again, what a topic that probably is more appropriate towards the end of the course. Okay, so that's the specification, which I've run through. Clearly, where you go from there is, right, well, where do I start and how can I do it? And so Pearson have produced schemes of work to help you. And the schemes of work are written in the same order as the specification, which you might or might not, to be honest, probably might not want to teach in exactly that same order. But nevertheless, that's how it's been written for you. And one of the big advantages of them is the resources that are being put in. So the schemes of work are available from that download page that I showed you at the beginning of today. You can just take them. And one of the key benefits of them, if we look at the next slide, is you can see there are hyperlinks in there with the different connections and giving you ideas as to where to find resources. So if I just go back for a minute. So the schemes of work are available as a Word document rather than PDF, so you can cut and paste and edit to them more easily. There's worked on the basis of about 120 to 140 guided learning hours. In other words, that's ignoring homework and material like that. Guided learning hours would be you in front of your students. So if you want to break that down over term time, over 2 years, it gives you the sort of chance to start planning how to cover most of the spec. There's course planners, there's lesson plans, there's suggested approaches, and it's working on about 2 hours of teaching time per week over about 60 weeks for the course to be completed. There are getting started guides and obviously units like what the work we've done today, which will help you sort of with that structure. So as I showed you earlier, here's a sample page of those schemes of work, which, as you can see, gives you the basic spec content, then breaks it down to specific spec points, looks at the sort of things you could do, the exemplar activities with links to various resources, links to the exemplar materials and other resources. We talked earlier about transferable skills, so it shows you, what's available with transferable skills that can be used as part of that. So those are the schemes of work. So let's sort of have a little break from me talking for a minute. So let's just think about the order of the topics that you would teach. Okay. So if you'd like to put in the text box, fill in the text box to list 3 topics that you think should be covered at the start of the course. Let's do that first. Let's just do that first bullet point first. So what topics do you think should be taught at the start of the course? Put your ideas in the spec box. You can be quite general. So if you think, you teach topic 1 first and go through the sort of classification. What about teaching diffusion and osmosis early on? Because that understanding of how things move. Yeah, cell structure, biomolecules has been suggested in there. I certainly agree with that. Yeah, characteristics of living things. Cells, absolutely, totally agree there with Annabelle. Cells, variety of living things. The cells and organisms coming up again, microorganisms. Don't forget some of the basic core practical things as well. Things like food tests can be a nice, fun, practical to do early on, but teaching basic sort of skills in terms of, lab, wear, safety, heating. They can be useful topics as well. So let's move on to the second one then. So just to make the sort of chat box a little bit easier, what sort of things do you think you should leave to the end of the topic? So make some suggestions in chat now to the things that you should teach towards the end of the spec. Yeah, and sort of suggest, unit 5 sort of stuff. Yeah, micro cycles, nitrogen cycle, carbon cycle. You can't really understand what's going on if you haven't covered diffusion and osmosis and basic sort of biological molecules. A lot of people suggesting leaving some of the genetics till later on. Again, sort of an understanding. If you haven't done cell structure and all the rest of it, then mitosis. You're not going to understand mitosis if you haven't covered what chromosomes are and what they do. So yes, I'm getting lots of genetic engineering, biotechnology coming towards the end of the course, I'd agree. And what about the third bullet point then? Are there some topics which are synoptic, topics which require you to pull together other bits, which might be very useful leaving to the end, but also perhaps spending a bit longer on them so that you sort of revise some of the stuff that's gone before and then develop them further on. So yes, and exactly, yeah, I'd agree with Veronica there, immunity, and that brings in a lot of different things to do there. Yeah, fertilizers, Alia's sort of saying the use of fertilizers and nutrients, if you can't understand how they're absorbed or what functions they are in terms of the nutrition. And yes, Chara is suggesting eutrophication. To me personally, in my teaching, I think eutrophication is one of the classics. And it lends itself very well to exam questions because there's an order to it, the understanding of what's happening, the build-up of nutrients in the water, the effect that has on the algae, the increase in growth on the algae, the effect that has on blocking out light, the effect that has on bacteria, and so on. So yes, key variation that Ahmed is suggesting there, definitely topics. And the whole environmental thing, Chibodi is suggesting, the environment. I would agree with that as well, to draw together lots of ideas there. So I think what we're saying is that it's not just a matter of sort of picking up the schemes of work or the specification and starting at page 1 and going through it. So the schemes of work are deliberately written like that in order to help you find things. So they're in the same order as the spec. But as I said earlier, they are published in Word, so you are capable of actually, cutting them up and cutting and pasting and using them, and putting them into your own schemes of work, your own order. obviously in the end, you don't want to be having a mad rush saying, oh my God, we haven't covered sort of, the whole of topic 5 and it's a mad panic because the exam's 2 weeks away. So having that sort of structure as you go all the way through. I think this next bullet point number 4 there is really important. That ability to revisit themes, being able to start your lesson by saying, remember when we did so and so, so and so. Let's just refresh our memories again. What was diffusion? What does that mean? What was this concept of concentration gradient? What's a semi-permeable membrane? Right, okay, now that we've done that then, so we're looking at the lungs today. So what are we going to have going on in the lungs? Well, we're going to have oxygen coming in and we've somehow got to get it into the blood and we've got to get rid of carbon dioxide. So how is that going to work with concentration gradients? So you're pulling things together by revisiting one topic before you introduce the next. And again, one of the things I didn't see come up, actually, I didn't think about myself, that's mentioned there, fish farming. The implications of putting a large number of sort of salmon together somewhere in a big net and feeding them artificially. Where does all that disease goes? What happens to the waste? And so on when you're doing that. So some sort of key ideas there. Yeah, not sure where these red lines have suddenly appeared for. Apologies if you for those. So underpinning topics need covering early. So the cells, the transport that we've been talking about there. Some topics are often found to be more difficult. Students get confused between transcription and translation. So it's perhaps better to place them towards the end of the course, as long as you're confident that you're not going to be rushing them at the last minute. Some topics require knowledge from the other areas of the course. So things, as we mentioned today, like gas exchange and diffusion. Some things draw things together, so you can use them that way around, the digestion, nitrogen cycle, energy flow. And then, of course, when you do practicals, if you're going to go outside and throw a few quadrats around and look at the ecology, then you may want to do that at a time of year when it's more pleasant to be outside and when you can find more interesting stuff on the ground. So it may be that some of the other practicals, there's availability of resources that will affect when you can do them. So there would be further considerations in terms of that. So yeah, so I hope that's given you a chance to sort of think about the job in hand of getting through that specification over whatever period of time you've got, whatever resources you've got, how many students you have, what backup you have in terms of practical and IT support in order to develop that course through. But you've got the core materials there from Pearson that you can cut and paste and chop up and use, and you're not on your own. There's a lot of material there to help you, all available from that Pearson qualification website, which again, I'll put up later. So let's just look at another sort of activity for a minute. So if we're talking about one thing linking to another, to a certain extent, we've covered this, so I won't spend too long on this. But, here's some spec points, 246 through to 250. Describe the structure of the thorax, including the ribs, intercostal muscles, diaphragm, trachea, bronchioles, alveoli, and the pleural membranes. So again, in terms of the spec, very specific for you, you might forget about pleural membranes. You might forget about bronchioles, but the spec is saying, no, there could be a question that comes up where there's a diagram of the lungs and they're asked to label the trachea and perhaps 2 other things. And if you haven't covered bronchioles, they're going to find that a little bit harder to do. The role of intercostal muscles and the diaphragm in ventilation. One of the classics there is the bell jar experiment with the rubber diaphragm and the balloons. And you can talk about how that has limitations in terms of the absence of intercostal muscles, but the diaphragm is working on pressure and ventilation, and how that can affect people that are stabbed or have got a broken rib and it can affect how the diaphragm ventilates the lungs. The alveoli and gas exchange by diffusion, understand the biological consequences of smoking, and then a practical investigating breathing in humans and the release of carbon dioxide. So the thing I'm asking you to put in the spec is what other topics would come along with that. And clearly the obvious one, you know, which I'm seeing a lot of people putting in there, Darshan's put it in, you know, diffusion. Then why you're bothering with all of this? Respiration. And then what you're going to do with it when it's into the lungs, transport. So yes, you in there, I absolutely agree with you. Hit the three spot on, really. Diffusion, respiration, transport, movement of substances, you know, that's it. So that understanding, I think, is important when it comes to our teaching, that we don't just see things in isolation, that we sort of put them together to take them through with what we're teaching. And again, you know, you could do the same thing again here. You know, that's what we're doing. Like spec point 215, which came earlier, diffusion, osmosis. Quite a few of you said that. Understanding the effects of the rate of movement of substances. So, you know, surface area to volume ratio, that would come in there. The classic experiments, you know, put potato chips, things like that, on diffusion and osmosis. And then understanding what you do with it after you've done it, with the respiration. So, you know, the understanding of adenosine triphosphate, the ATP, the differences between aerobic and anaerobic respiration. That's, for some reason, students get very confused with that. I don't know whether it's just because the words sound the same, but understanding that principle of, you know, the buildup of lactic acid and so on with anaerobic respiration is an important part to bring in there. And again, there's another chance for another experiment there with respiring seeds. So that might affect, you know, the time of year when you do that. You perhaps towards the end of a term is not a good time to set up experiments that have got seeds have got to germinate. And you've got to come back and look at them, and perhaps a week later or 2 weeks later to see what's going on with the kids' experiments. So factors that affect what you teach and when you teach it. Again, you know, there's where diffusion came up again. And then although all the flowering plant stuff is in bold, so it would only be on paper too. Again, you know, sometimes we've been concentrating just on humans, but understanding the role of stomata in gas exchange, the difference between day and night in plants comes in there as well. So really what I've tried to do with you there is just to help you sort of get your head around the cross-fertilization, if that's not too much of a pun in biology, of the different topics and how you can use one topic to reinforce another. And that's important in the students' teaching. You don't just do, right, we're taught this, we've done it, forget it till it comes to the exam and revision. We now do this, teach it, do it, forget it till it comes around to revision for the exam. You're constantly sort of linking across how one topic is related to another topic, how one information that we learned in a previous lesson is important to our understanding of what we're doing now. And this idea is a word that's used there, this metacognition of getting to evaluate their own knowledge and saying, oh, yeah, I'd forgotten about that. Or, you know, I need to sort of go back to that and check it again. And we can use various techniques, which I can sort of perhaps describe to you now in terms of how to support students to do this. So this is one way of doing it. When you've sort of taught a topic or something, or you've given them a test, or you're starting something new, you could give them the spec points here, and you can see the emojis at the end. So you say to the student, right, what are you going to do? So know the process of respiration releases energy in living organisms, understanding that energy can't be created or destroyed. It has to be changed from one part to another, the importance of glucose, etc. How well do I understand that? Am I going to give it a smiley face because I'm very confident? Am I going to give it a straight face because I'm not sure? Or I'm going to give it a sad face because I've forgotten all about that. I don't remember you teaching it to me, and I didn't understand it even when you did. So you can give the students an opportunity, and obviously if they all end up with their ticks in one page or another, then you do it. I think someone's asking, where are these? I'm not sure we've produced these. I think it's just an idea for you to develop. So you could obviously just copy the spec and then ask the students to, on a bit of paper, to give it a score of 1 out of 3, or copy it into a spreadsheet and get them to do it that way around, and then cut and paste up the different bits as you go. So it's just a way of sort of the students having a chance to say, this is what I should have learned. How happy am I with this? And you could use this at revision. So you could have a lesson on revision of these topics. This is all around respiration, ATP and everything. And then the start of next lesson, you could go back and say, right, okay, what last lesson we revised respiration. Here's the key topics that I thought we'd covered. How do you feel about them? Do you think you've got them? Have you not got them? And that could be the start of the next lesson after you've just revised these topics. So it's just an idea to sort of float with you in terms of letting the children see the spec and get them a chance to do it. And don't forget the command words there, like explain, understand, know, the things that you should do. Another different way of doing it is like this. So here, and we'll talk about these AO1, AO2, AO3 in a minute. Here you can talk about a way of after the exam. So you can say, right, okay, you've just done a mock exam, or I've just given you an internal assessment, and let's look at question 1. Well, question 1, the first part of the question was on spec reference 2.54, and you could look that up. You can get the students to do it, and you can say, right, okay, spec point 2.54. Let's have a look at that. What was it? 2.54 is describe the role of enzymes in transporting water and mineral ions from the roots to the plant. Okay, that's what that question was about. It was worth 3 marks. What did you get? So write in your score, get your exam paper. I've marked it for you. I've given it back to you. Don't just look at it and say, oh, I got 72%. Drill down to question by question. So you got 2 out of 3. Why do you think you didn't get 3 out of 3? What did you miss? And I'll talk about AO1, AO2, and AO3 in a minute. This particular one is a recall of knowledge, so it's AO1. Then look at question part 2. That's on the same spec point. We're still on xylem and so on. So what about that one? How many marks did you get out of the 2? Then it went on to question 1B. That was the transpiration bit and the understanding of what transpiration is. So how many did you get for that? And so on down. Then you can look at the total for that question. So, right, okay, you could have scored 19 marks on question 1. How many did you score? Now, is there a pattern? Did you get more of AO1 wrong or right? Did you get more of AO2 wrong or right? Did you get more of AO? So that might help them to sort of say, well, I'm getting the knowledge, I'm getting AO1 questions right, but I'm not applying them. It's AO2s that I'm struggling with. Or I didn't think this practical was that important. So it's the AO3 marks that I've drifted out. So this gives you a chance to sort of, when you've done a past paper with the students, rather than them just saying, oh, how did I do? Oh, I got so-and-so %. What grade would that be? That would be a grade 7. Oh, well done. Okay, I'm happy with that. Thank you very much. Put it in the bin, move on. No, drill into it, question by question, and analyze which questions they were good at, what style of questions they were good at. Did they struggle with explain questions? Did they just lack knowledge? Did they make silly mistakes? When it came to multiple choice, did they rush into the first item and not consider that the other 3 were definitely wrong? And so they made a silly mistake. So this is one way of actually, using a past paper that you've done with them, instead of just giving them an overall score, giving them an opportunity to drill down and understand which question types they're getting wrong and which question types they're struggling with. So what were these assessment objectives that we were talking about? So you have assessment assessment 1, assessment 2, and assessment objective 3. The questions on the exam papers will always cover all 3. We have strict rules, which I'll show you now, about the amount of each type of question. And some teachers put a lot of emphasis on this and try and analyze what the students are doing. Others see it as just a sort of a balance, and it's not so important to focus on in their teaching. But basically, if your students are thinking, well, all I have to do is learn the facts, they're going to be disappointed, because only 40% of the questions are AO1. And AO1 is knowledge and understanding. What is the name of this? What is that? A fact. And just regurgitating facts will only get you 40% of the marks. AO2 is that application of knowledge and understanding, analyzing and evaluating what's going on. And often students find them more challenging. Okay, so if there's I don't know, I'm making this up. If there's a picture of a donkey and there's a question about the donkey, they don't go, oh, we never did donkeys. I can't do this question. My teacher's terrible. They know that 40% of the questions are going to be stuff which they won't automatically recognize, but they have to apply their knowledge to a new situation, a table of results, an article about eutrophication or something, the story of what's going on in a lake. They have to pick out from it. This is where the transferable skills come in. They're having to pick out from the information that's presented to them and then apply the knowledge. And that's another 40%. And then AO3 is the practical. So 20% of the marks are on the practical. And as I said to you earlier, the best way of teaching the practical is to do it for real, hands-on, and to discuss it afterwards. And one of the best things I think can happen is when the kids turn around and say, my experiment didn't work. Because then you've got the opportunity of saying, why didn't it work? What was going on? Did we have too much concentration of a certain chemical? Did we not have enough of a certain chemical? Was it too hot? Was it too cold? What would be the impact if it was too hot? Did the enzymes denature? Did you not warm it up enough? What would be the impact if it was too cold? All of these sort of things can be used in teaching as far as the practical is concerned. It's not just a matter of, it's not cooking. You're not just following a recipe and doing what it says. You're experimenting, you're investigating what's going on and trying to understand the results that you get. So just looking at those in a little bit more detail then. So AO1, as I said, is knowledge and understanding. Okay. They can feel confident. I can label a diagram of the eye. I can label a diagram of the lungs. I know the words trachea, bronchus, bronchioles, alveoli. That's straight AO1 questions. It's easy on revision. And so you can use things like repetition. One of the techniques you can do, which I found quite good, is you say to students, right, okay, the person that you're sitting next to, decide who's student A and who's student B. So they go, oh, I'll be A. They get used to it when they've done it for a while. You say, right, student A, explain to student B the structure of the lungs. So student A has to explain to student B, and then you say, right, student B, do you think that was, how well did they do it? What did they forget? And then you can go on to, right, so student B, now explain what the diaphragm is. And the kids get used to discussing things. They get used to not being embarrassed. They say, oh, I've forgotten that. What was it again? And you can, teaching, I found, was the best way of learning. So getting the kids to teach each other can be a very good way, especially with AO1 about the facts. Mind maps are another way of doing it. Flashcards, turning over the flashcard, what's the name of this? multiple choice questions can all be techniques for developing AO1 skills. So here's some typical AO1 questions, different styles of questions, but still all AO1. So basically what it's saying is, name the organ that produces bile. 1 mark, 1 right answer. The answer is liver. There's no alternatives. They know it or they don't know it. But multiple choice. Multiple choice, a lot of people think multiple choice are easy. Well, they're easy in that you've got a 1 in 4 chance of guessing. But they're not easy necessarily from a science point of view, from an understanding point of view. But the technique on how you answer them should be sort of, which organ releases progesterone? The brain. Oh yeah, it'll be the brain. A. I never read the rest. I didn't think about it. So I need to sort of go, right, okay, which organ releases progesterone? Now, what is progesterone? Okay, it's a hormone. Now, what was it? Oh, testosterone is the one in men. Progesterone, estrogen, progesterone, that's a female one, isn't it? So it could be the brain or the ovary, because it's a female one. Pituitary, well, that's inside the brain. Testis, it's not going to be testis because that's a female hormone. Right, I've got rid of 1. So now if I'm struggling, it's not 1 out of 4, it's 1 out of 3. And so you can drill down like that with a multiple choice question. And it's good to teach the kids to think that way on a multiple choice question. Then, of course, another style of multiple choice is based on the table. So you've got to be able to systematically go, right, which row of the table correctly shows whether the kidneys and the skin, yes, there's 2 columns there, kidneys, skin, excretion. So have the kidneys got anything to do with excretion? Yeah, they have. Has the skin got anything to do with excretion? Yeah, that's sweating and stuff like that. I remember my teacher doing that. Right, okay. So it must be, they're both involved. Okay, so where's that in the table? Well, it's not the first one. It must be the last one, D. So I didn't just rush to the table. I didn't take a guess. I thought about the question first, thought what I thought the right answer was, and then searched for the right answer within the question. And that can be a better way of tackling these type of questions and doing that with the students. And, do a couple of multiple choice questions at the start of every lesson. This is a fun way of sort of getting them to think about what they did last time. So AO2, as I say, can be more challenging because they have to apply their knowledge. There may be a familiar situation, in which case it becomes a bit closer to AO1. Sometimes when you ask about a core practical, it's difficult to decide, is it AO1 because they're just remembering what they did? Is it AO2 because they're applying their knowledge to some different sets of results? It wasn't the results they got. Or is it AO3 because we're asking about the practical techniques? So these criteria can be debatable sometimes, but the AO2 is about applying your knowledge. Okay. So it could require calculations. There could be quantities involved. It could be quantitative. So calculations, graphs, analyzing tables and data and so on. It can require higher skills. So questions like evaluate and discuss. It's important in those sort of questions where they give both sides of the argument. It could be this, it could be that. Between the two, this one seems more likely. That's a sort of thinking when it comes to evaluate and discuss. As I said to you there, the initial reaction of the students might be, I don't recognize this, you never taught me this. But they have to expect that because they know 40% of the paper will be about applying their knowledge. And if they see the command word suggest, then they're going to be thinking, well, no, there isn't one absolute, definite right or wrong answer. They're going to, I've got to come up with some suggestions on this. So here's a typical AO2 question. Heart disease is a major risk of health in the United Kingdom, right? That's just an introduction. I knew that. That's nothing funny. In a study, the number of people with heart disease was recorded. Right, okay. The table shows the results of the study. Right, I'm not going to look at the question yet. I'm going to get that table into my head. What's it telling me? Age, first of all, you'll notice this is question B. So there was a part A before this feeding in. It's not just suddenly out of the blue, but that's not shown here. So what's the table showing me? So I've got age ranges, and then I've got number of people. Now, what are the units? What's this all about? Number of people with heart disease per 1,000 of the population, right? Okay. And then it's split between males and females, right? So what's going on? So in every case, 5, 3, 138, 118, 305, 200, in every case, the male is higher. And in every case, the numbers are going up as you get older. Okay, is it telling me anything else? Don't know. Right, what's the question? Give two conclusions that can be made. Well, I already have. It's higher in males and it goes up as you get older. Write those down, 2 marks, job done. So you can see how what I didn't do is read the question and then go back to find the answer. I got the information into my head first, I understood it, then I went to look at the question. And that's about applying your knowledge. Okay. AO3 then is the practical. So here we're quite clearly just based on the core practicals, although you could be given an experiment which they haven't seen, and there could be an element of AO2 in there, that they have to devise an experiment that would work for that particular challenge that they've been given. So words like variables, accuracy, reliability, evaluating, data are important words to be able to get in there. So they can be quite challenging. So here's a question. The table shows the results the student obtained from her investigation. Again, this is part B, so it's not suddenly out of the blue. There was a part A before this. So it's obviously something to do with shining different colored lights on a plant and counting the number of bubbles released. And one of the things blank. So complete a table by calculating the average rate for red light. Okay, so let me just check. So average, you add the 3 together and divide by 3. So is that how they got 19? Yeah, that works. Yeah, so I need to add together 23, 26, and 25. I'll write down that total just in case I make a silly mistake, and then divide it by 3, and then put my answer in the box. Do I put it in the box? Complete the table. Yes, even though there's an answer line there, it says complete the table. So I'll put my answer in the box, and if I put it on the line, it won't matter. The teacher will still mark it because the line's there. And then I've got an explain question. Explain whether the results for each color are reliable. Okay, so that's really testing my understanding of practical and this idea of reliability. Well, the reds are going through a range of 3 on really, 25, 26. The blues are fairly close. Ooh, that's 6 on the green. Looks a bit funny, doesn't it? Is that average of 14 reliable? Oh, hang on a minute. How did they get that 14? 14 is 12, 28 divided by, so they ignored that 6 to get the 14, right? That's what this question's all about. So green is not reliable. Why? Because in trial 3, the result was so far out, I missed it out in terms of my average. So you can drill in like that with this type of AO3 question. Here's another AO3 where it takes you right back. Plant growth substances stimulate root growth from a cut stem. Perhaps the word auxins is coming into my head. Describe an investigation to find the best concentration of plant growth substance to stimulate growth. You should include experimental details in your answer. So here's a describe an investigation, not necessarily one I've done before. So I'm clearly going to have to take a certain number of plant cuttings. I'm going to score marks for saying whether or not they're all from the same plant, the same size, the same length, then keeping them in the same conditions. So the variable is the amount of auxin I'm putting on each root, or the concentration of the auxin in the fluid that are sitting in. And so those would be the things that would score me marks or not score me marks when I was looking at that. So let's get you to do something again rather than just listening to me. So again, if you can tell me, what AO do you think these questions are? So here's a question. Do you think that's AO1, AO2, or AO3? You put it in the chat box. So do you think this question is AO1, AO2, or AO3? It's interesting. We're getting a full range. We're getting AO1s, AO2s, and AO3s. Let's see a few more. So is it straight knowledge? Is it asking you something I've learned? Well, you could argue perhaps because have I learned what a dependent variable is? But then I've got to apply it. So no, I don't think it's AO1. AO2, is it, again, I'm not applying my knowledge to this particular situation. I have to understand what a dependent variable is. And a dependent variable is a practical skill. I think this is a practical question. I think it's AO3. So I can fully understand the debate, and quite a lot of you went for AO3. Don't think just because it's multiple choice, it's AO1. Okay? But ultimately, it's about practical. It's all about dependent variables, which is a practical skill, and say that particular question was classed as AO3. So say some of them are debatable, but that was classed on the exam when that came up. The examiner classed that as AO3, and I agree with them. What about this one? Pollution can occur in the atmosphere and in rivers. Carbon monoxide can pollute the atmosphere. Describe the effects of carbon monoxide pollution on humans. So I think quite rightly, nobody's going for AO3. It's clearly not a practical question, so I'd agree with that. So am I applying my knowledge to a situation I haven't seen before, or am I just describing knowledge that I have about the effects of carbon monoxide pollution? Again, I can see the debate, but that one was classified as AO1 because you basically had to know what carbon dioxide did or not. So it was recalling the effects of carbon monoxide. And this one, if the mineral ions are not absorbed, this is part C, so obviously stuff came before this. If the mineral ions are not absorbed, they are egested in the feces. The feces of genetically modified farm animals contains lost phosphorus in the feces of normal farm animals. Some people catch fish from rivers near the land. So quite a lot of context there. Discuss, as a command word, why these people might support genetic. Yeah, and everybody's got this one. Yeah. So AO2, because you're very much, when it's discussed, it can't be AO1. There's no practical context there, so it can't be AO3. So it's clearly AO2, which is nearly everybody has gone for. Excellent. Well done. So I've been referring to these sort of command words as we've gone through. So the command word is a word at the start of the question, and it's important that the students understand them. And in the specification, if I remember rightly, it's Appendix 5 or something at the back. It lists all the command words. Yes, it is. It's Appendix 5. I would give the students this. I would print this off and give it to them and stress the importance of understanding the command words. It has been updated just slightly. It hasn't been framed, but we can use the word compare, and we can use words like write or measure. Just, technically they should be in there as well, but I don't think they're anything significantly different. One thing that is the most common mistake is the difference between describe and explain. If, for instance, I gave you a question which said, here's a graph, describe what you can see on the graph. A simple answer would be, it goes up, levels off, and comes down again. A better answer would be, it goes up to 27, levels off till 36, and comes down to 50. That's a describe. An explain would be, it goes up to 27 because, and that's a because that makes it the difference of an explain and a describe. It goes up to 27 because as the temperature rises, the activity increases. It stops at 60 degrees because the enzyme was denatured. And in the mark scheme on an explain question, the second part of the marks at least will be on that part. It'll be on the why did this happen? And use what I've just done with you. Say to the kids in an explain answer, where is your because? Where are you explaining why this happened? You're not just describing. And if you're asked to describe two things, then surprisingly, you need to describe two things. You don't have to explain them. You have to describe two things. So if you in a describe question go, this happened because, you'd only get 1 mark because you're asked to describe two things. So understanding command words is very important to exam technique. There's an excellent definition of every one. As I say, I would take that out of spec and give it to the kids. Core practicals. So we've mentioned the core practicals when we went through the spec. So they all, they're there in the spec. They can be tested on their knowledge of how they did them and the results that they got. They can be put into sort of other contexts. So for instance, you might have done food tests and tested for starch, but you could be asked about testing something else for starch. You could be testing a blood sample for glucose, even though you did it as a food test. So it can come in a different sort of context. And as I say, the best way is for the kids to actually do them. They have to be aware of the sort of safety procedures. Would you heat it? Would you use a water bath? Those are important parts. Students should understand how to explain the experiments in order to get valid, keyword, to get valid data. And students that understand how to analyze and evaluate the quality of that data. And there's all the core practicals. You can see some of them are paper 2 only. So if you're doing linear and you're coming to paper 2, then I would make sure those 1, 2, 3, 4, 5 core practicals have definitely been revised, because there's a good chance or a higher chance that they will be there on the paper 2, and don't bother revising them for the paper 1, because they won't be on the paper 1 because they're in bold. So the core practicals are listed there for you. And here's a typical core practical question. Okay, so this is obviously investigating diffusion. So again, if I was a student, I'd be saying, right, this apparatus can be used to show osmosis. Ah, yes, I've done this. I recognize it. It's one of the practicals we did. That's great. Okay, right, but what's going on? I've got a glass tube. The sucrose solution is the one that's in gray. It's sitting in water with a partially permeable membrane. Right, so what did my teacher tell me? Yeah, what's going to happen? The water is going to go in through the partial permeable membrane, and that'll make the level rise. Yeah, I've done this experiment before. I recognize it. I've worked it out. Right, what's the question? Explain, command word, explain what happens to the level of sucrose solution in the glass tube, right? Well, I've just thought that through in my head when I was doing it, but it's an explain question. So I might get 1 mark for saying it goes up. But then I've got to say, because I've got to explain why it goes up, because it's an explain question. Describe how this apparatus could be modified to measure the temperature at different temperatures. Oh, okay. So then I've got to apply my knowledge now to this practical situation. How could I do that? Well, could I set it up with beakers where the water's at different temperatures? How many would I have to do that? Would I need 5? Would I need 10? Would I need 3? Would I need to repeat it to check that my results were valid? So implications come up there in terms of part B of that question. Okay, so student, and here's another one here. Now again, what do I get into my head while I'm doing it? The student investigates the oxygen absorbed by germinating seeds at different temperatures. Okay, what's going on here? The diagram shows a student's apparatus. Okay, I think I've seen this before. I think my teacher showed it to me, or perhaps I haven't. So I've got to try and work out what's going on. So soda lime, do I know what soda lime does? I've got germinating seeds. So germinating means they're growing. And I did mark this question actually, or a very similar question. And I got loads of answers about bacteria and from weaker students. And I didn't understand why they'd got it from. Then I eventually twigged they didn't understand the word germinating. And it was germ. And because it was germinating, they read germ as bacteria and started writing about bacteria. So that just shows the importance of the vocabulary and understanding what's going on. Okay, so here we've got an experiment where I need to understand what soda lime does. It's in the spec, the role of soda lime. It's absorbing the carbon dioxide. That's going to reduce the volume. So the colored liquid bubble is going to move to the left. So what's all that tap and syringe all about at the top? Well, that must be about rebalancing the pressure again. And that's going to set the bubble in the scale. Okay, so what's the questions all about? Suggest why the student opens the tap after obtaining 1 set of results. Right, well, I've just done that. And then what's the function of soda? Right, both of those questions I can now answer just as I was thinking through what was going on. And you can see the spec points that that question was related to. Okay, so the experimental skills, you know, here's a list of the sort of skills behind them that it's important that they need to do. To help you with those, here are 2 booklets, which are again available from that website that I showed you at the start. So you can go into Pearson qualifications, support materials, and there's a guidance on practical terminology, and there's a core practical guide. This covers biology, chemistry, and physics. So it goes through practical by practical. The practical terminology is very useful when it comes to sort of describing the thing. So it just literally, it's not a big document, it's only a couple of pages, but it sort of talks about it, but it in bold stresses the words like scale, it stresses words like range, repeat, precision, reliability, line of best fit, anomalous, results. It goes through all of those and explains what they mean. So that's a guide to using practical terminology. And then the other one is a much bigger document. As I say, it's about 100 odd pages, I think, because it does all the biology, chemistry, and physics. But as far as we're concerned today, it literally goes through each of the core practicals, gives you the links of the spec points that are related to it, and then gives you sample questions. Excuse me, gives you sample questions that you might get on those core practicals. So 2 very, very useful documents that are worth having when it comes to the teaching of the core practicals. Excuse me a minute, sorry. Okay, so the other important thing, as I mentioned earlier to you, was the math skills. talk to your chemistry teachers, talk to your physics teachers, talk to your maths teachers about the math skills that are required. And just like we had for the command words, in the spec is a list of all the math skills that are needed. So again, you could use these with your students. The BCP there with the ticks is biology, chemistry, and physics. So you can see many of them are common to all three sciences. So if your students are doing triple science, if they're doing biology and chemistry and physics, then talk to your chemistry teachers and talk to your physics teachers and be clear how these things are being taught and what they need to know. You'll see very few of them, if any, are biology only. So there are core concepts that they need to develop as they go through the course. And here's a typical question. Now, again, you can see this is part 2 of a question that's had more introduction. It wasn't just suddenly thrown at them. So they've got the data there that they need to sort of work through and do a maths calculation. It's important that they always show they're working. If they put their answer on the answer line and it is correct, they will get the full 2 marks with nothing else there. They won't be penalized for not showing their marks. But if they put the wrong answer on the answer line, they will get 0. If they put the wrong answer on the answer line, but there's working there, the teacher, the examiner, can look at their working and the mark scheme will show that they might get 1 of the 2 marks because they, for instance, correctly halved the population, because it says the population is 65 million, of which half are male. Calculate the number of males. So if they successfully halved 65 million in their working, then it may give them 1 mark. Not saying I'm guaranteed to it, it obviously depends on the mark scheme. So that has to be done as you go through. So look out for maths questions, practice maths questions, share the teaching of maths questions with your colleagues in biology and chemistry and physics. And just like the core practicals, there is a maths guidebook, again, downloadable from the same page. And that goes through exactly as, you know, when we're talking here of the different math skills, then it goes through those skills one by one in the book and explains what's required, what is the approach to doing it, worked out examples, sample questions, and so on. So a very useful document. Again, it covers biology and chemistry and physics. So if you're just doing biology, there will be some of it that's not appropriate because it's straight science all the way through. But you can pick and choose the bits out, and particularly sort of, you know, things like graphs and scatter graphs and lines of best fit, proportionality, gradients, correlations. Pie charts could well be used. Certainly block diagrams, and when is it appropriate to use histograms or block diagrams. That sort of information is all there in that guide. Again, downloadable from the Pearson qualification website. Okay, so just summarizing then, we're getting towards the end now of the support that's available to you. So most of this we've discussed as we've gone through. So you've got, you know, the getting started guides, courses like this with training events, either face-to-face or online. Most countries have subject advisors, as well as the arena, who I'll show you the details at the end, for the Pearson direct advisor. But I was lucky enough to be out in India and Sri Lanka doing some training, and I met some of the sort of local Pearson reps there when we were doing that sort of face-to-face training. Certainly before COVID, although I think there are still some face-to-face training events. The schemes of work we've talked about, the idea of skills mapping, that one I showed you with the smiley faces and having the box after their exams. The loads and loads of sample assessment materials. Now that there's sort of 2 or 3 exams every year, and this course has been going for over 5 years. There's an huge amount of past papers available to you now. Exemplar mark responses. I'll talk about Exam Wizard and Results Plus in just a minute. So there's the website again, and you can see, that the number on that has changed. It's now saying 123 exam materials are available. If you remember at the start, it was just I think it was 3 on the first slide that I showed you. So it's obviously much better now to be able to follow that through. And that number now is much higher than 120 the last time I looked. The teaching and learning materials, 40 available. That's where you'll find the schemes of work, the guides. There's also some specific subject guides, which I'll mention in just a second, that are available there as well. So that is your key page to find when it comes to downloadable resources. So there's the booklets that we've talked about already, the maths ones, the core science, the practical guidance, but there's also, you can see that one there, I think there's 3. That one there that's illustrated is cell structure and stem cells. That's basically the slightly more challenging topics. There's support booklets on. There's another one on genetic modification and another one on protein synthesis. So there's 3 in total of those sort of topic guides. And then, as I said, there's summaries of courses like this in the Getting Started guide. So all those booklets are downloadable from there. Results Plus is very accessible. You can see the hyperlink at the bottom there, and you've got a copy of these slides in resources, so you'll be able to click on that directly yourself and go to Results Plus if you haven't used it already. In there, there's there's YouTube videos that direct you how to go through it. Effectively, what Results Plus is all about is after the kids have done their exams. So you can do mock exams and load them up, but it's most effective, as the name suggests, in the analysis of the results. So you can put in various search tools. You can compare your center's results to other centers of similar types. You can compare your center results this year to last year. You can go into individual student papers and look at them and get it. Basically, it's an open, all the information from the exams is open and in there, free of charge, for you to go in and have a look around and have a look at information. Primarily, as I say, after the kids have sat their exams, so that it's populated with the data that's relevant for you. So that's Results Plus. As I say, it's very straightforward to access. You can see in the last page there above number 4, what the pages look back. You basically have to put in your search criteria and you find it. Because what happens when papers are marked now, the days when the exam papers were all parceled up and the examiner's got a big sort of parcel arrived to mark them have gone. So when the students sit the exams, the papers are scanned, the questions are then divided up, and things like multiple choice questions are marked by a computer. Single word answers would be marked by some people. The more difficult explained questions would be marked by a different set of people. So all these are done, and then all the things come together, which means there's an electronic record of the kids' mark on every single item as it goes through. And you're able, therefore, to be able to go in and drill through that. And you could do the sort of analysis that I showed you earlier. You could see if they get AO1 questions wrong or AO2 questions or the style. Exam Wizard is effectively a database of all the exam papers. So you don't have to go to a cupboard and sort of get, a copy of each paper out from the bottom of the cupboard. Exam Wizard is a database of all the past papers, and you can go in there and search. So if you've just taught eutrophication, you can do a search on eutrophication, and it'll come up and give you a whole set of questions on eutrophication. If you want to do a test on a certain part of the spec, you could search on just that part of the spec. You could search on AO1 or AO2 or AO3. So it's a search engine through all the past papers, and then you can stitch together what you've accumulated. So you can actually produce an exam paper in Exam Wizard, which are all AO1 questions, or are all topic 3 questions, or any other combination that you want. Or of course, you may just want to access a whole past paper. So again, it's free of charge. It's, you know, you can find it by just searching Exam Wizard on the Pearson website. Go into it, learn how to use it, get familiar with your way around it. And I'll mention this a bit more in module 2 next week. But a absolutely valuable way of you being able to do it. It means you don't have to download everything onto your own laptop, or you don't have to print things off and store them in cupboards. You've got it all there, accessible and free. Access to scripts really is probably the most useful thing when it comes to a student that perhaps says to you, oh, I'm really disappointed. I got a grade 6. I was really hoping for a grade 7. You can go in and access that student's script. You can access the marks. And if, for instance, and I'm making these numbers up, if you had to get 60% to get the grade they wanted, and you could look at it and you can say, well, 60% of the marks would be 120. Again, I'm just making this up. You scored 110. That's 10 marks away. You know, there's not much chance of thinking about a remark, because you're unlikely to find 10 more marks anywhere. Not unless we can look at it and think for some reason the last page hasn't been marked, which I think is impossible or highly unlikely anyway. But you could look for some reason why they did so badly. But if they're only 1 mark away, you could look through it and go, oh, well, yeah, actually, you know, you were given 3 for this question. I wonder why you didn't get 4, or I wonder why you didn't get 5. Yes, we could put your paper in for a remark. And then the school is not wasting its money. It's paying for it direct through that. So those sort of additional resources, there are sort of paid resources as well, and that the student textbooks and active books that come out of that. The actual textbook is available, you know, for schools to use, and you get access. When you've done that, you get a 3-year access to the active book. There's a code inside the textbook that gives you access to an online version of the book as well, if you bought one of those. The teaching hubs provide fully comprehensive planning and front-on class guidance, along with the exam preparation resources and CPD support to help deliver international GCSEs to a high standard. So you can access those as well. And that, you know, the schemes of work will obviously, as we said, be able to help you with less time on that. This is Irene, who is a Pearson support. So there's a telephone number there and an email address where she can go through and give it. So I think we're getting close to completion time. We've got 10 minutes left if we need it. I can see some people saying thank you, and that looks like they're probably moving on already. So that's fine. So otherwise, if you want to put any questions in chat, I'll do my best to pick them up. Otherwise, I'd just like to say thanks very much for attending.